Hi everyone. I hope everyone's doing well and midterms went okay for everyone. So how you get credit for the session is at the end of the session, I'll put up a Google form link and you will fill that out with your name, email, and your professor, and that's how I know who showed up for these sessions. Hello. All right, let's go ahead and get started. So today we'll be reviewing plant forms and plant structures. And if you guys have access to the worksheet, feel free to pull it out and follow along with me. So the first question is asking, how is the general morphology of various organs of plants correlated with functions of those organs? So in biology, there's been this trend about how structure matches function. So we're going to be looking at the same thing in plants. So let's discuss some structures of the plants and how it actually helps their functions. So one example that I can think about is that plants have thin, flat leaves. And what this does is that it's going to allow for high surface area to volume ratios. And this is going to be important because the greater the surface area is, the more sunlight will hit the leaf, and that'll give them um, enough light to do photosynthesis. Okay, then there's also structural advantages in stems in how their vascular tissues are arranged in a ring near the periphery of the stem. So can anyone tell me why having it in the periphery is helpful for the plant? So kind of think about why the stems are important to plants. Why do plants have stems? So it looks like Dr. Reddy didn't post this worksheet. So if anyone have access to the Google Doc, can you guys please put it in the chat so other students can access them? Okay, so stems have vascular tissue arranged in a ring near the periphery of the stem. So this is helpful because the stem of the plant are usually there to provide structure for the plant, right? So we want the stem to be really strong so that the plant doesn't fall over or fly off when there's like a really big wind. So having the strongest tissues, which are the xylem and the phloem, in the periphery rather than in the center is going to help them be very rigid. So that's why plants have their vascular tissues in the periphery of the stem. So I haven't put in the login sheet yet. I'll be linking that at the end of the session. I decided to do that from now on because if I put it earlier, people, if they come in a little bit late, they don't have access to it. So I thought it would be just better to put it at the end of the session. Okay, so we talked about the vascular tissues in the stem, and we see that this is kind of the opposite in roots. And if you think about it, roots need to be able to bend and it needs to like kind of travel through the soil, right? So we don't want something that does that to be very rigid. Otherwise, it's going to have limited mobility. So that's why it's opposites in roots. So we're going to have these strong tissues towards the center rather than the periphery. And it's able to bend much more. So I'm just going to write down that this is for the stem and this is going to be for the roots. Okay. Any questions on that first part? Okay, so now we're going to be looking at how water's property, like the adhesion and the cohesion properties of water, help maintain the flow of water in xylem and of the plant. So first, I can give a quick review. So cohesion is water molecules sticking to other water molecules. And adhesion is usually water molecules being attracted to like whatever it's in. So if it's in a water bottle container, it's the water that's attracted to the water bottle, and in the plant, it would be the xylem. Okay, can anyone tell me how these adhesion and cohesion properties actually help water flow through the xylem of the plant? Comment says they help move water upwards against gravity. Yes, they do. They do that. Can anyone tell me how it does that, how it's able to travel across against gravity. Another comment says creates tension which pulls the water up through the xylem. Yeah, so what the water molecules are doing is first they're going to kind of stick to each other. So each of the water molecules are sticking to the other water molecules. And then they're going to adhere to the sides of the xylem vessels, and they're also called the tracheids. So first the waters are sticking together, and then they're also sticking to the sides of the xylem. And because there's a negative pressure that is applied, it's going to pull the water up the xylem. And each water molecule is then therefore going to pull the next molecule with it because they're connected to each other. So it can kind of look like this. So the water molecules, so the water molecule here, let's say H2O, are being attached to one another, but then there's also going to be a kind of attachment to the xylem vessels. So this is the, and there's going to be a negative force, which is going to pull the water up. And as one water molecule gets pulled up, this water molecule also gets pulled up. Okay, so that's just like a illustration of that. I can also write it out. Okay, so we talked about how there's a negative pressure that's pulling the water up. So that means that water is going from a more positive water potential area to an area where there's more negative, which is like lower water potential, right? So so what do you think the water potential of the root is compared to the soil? If water is going from the soil to the root, then what must that say about the water pressures? So common says the root has a high water potential. So it would kind of be the opposite because the higher water potential goes to the lower water potential. So, oh, no worries. Yeah, the other way around. Yeah, so in a plant, the roots are getting the water from the soil, so the soil needs to have a higher water potential than the root, so the water can want to go to the root. So here's, oh, that looks like water. Let's see. So here's the soil. There's our plant. So the water is going to go from the soil to the root. Okay, and then another similar question. So what do we think about the water potential in the air compared to that in the leaves of the plant undergoing transpiration? So transpiration is the leaves evaporating their water molecules. So it's saying the air potential is low. Yes, that is exactly right. So if transpiration occurring, then air must have a lower water potential than leaves. So one of the comments says, does transpiration cause guttation? Like, is if the air is so low, that means if there is guttation, humidity must be raised. I'm not sure about what guttation exactly means, but if anyone else in the chat knows, please feel free to answer. Okay, so next we're going to be doing a practice problem with water potential. So this is the symbol for that. Okay, so a student uses a U tube for a series of experiments. Side A and B of the tube are separated by a membrane that is permeable to water, but not to sugar or starch. What results would you expect under the experimental conditions given below? Okay, so it looks like the solute pressure is always negative and 0.01 molar solution of any substance has the solute potential of this, and therefore 0.2 molar solution would have negative 23 times 2. Okay, so they're saying any solution with 0.1 molar solution is going to have this solute potential. So say instead of 0.2 molar, they gave us 0.5 molar, then we would just multiply that to get the value that we want. Okay, so here's the experiment. A solution of 10 grams of sucrose in 1,000 grams of water is added to side A. An equal volume of pure water is added to side B. What will happen to the concentration of water and sugar in the two sides over time? So first, we are going to figure out the components of the equation that's given above. So first we need solute. So they're telling us there's 10 grams of sugar or sucrose per 1,000 grams of water. And they're also telling us that the molecular weight is 3 142. So in order to get moles, what we can do is we can take the 10 grams and divide it by 342 grams per mole. And that's going to cancel this. So we're going to get 0.03 moles of sucrose. And it's still per 1,000 grams of water. So we're going to apply the principle that we learned about this solute pressure, how we can just multiply it by our moles to get our new value. So here we have 0.03. So what we can do is we can multiply. So to get this value, we can do 0.23 times 0.3. And that's going to give our new solute pressure, which is negative 0.069. So the pressure is 0, and then we just found the pressure of the solute, so that's 0.069. And then when we calculate that, it's going to make it positive. Or sorry, not positive, it's going to be the same. So the reason we put 0 here is because pure water is on the other side. I'll make a note of that here. Okay, so what's going to happen is, so we have a negative number. So from that, we know that water will move from this region to the higher water potential to the side with the sugar, which has the lower water potential. So negative 0.069 is lower, right? So the water will move from high potential to low potential. Okay, any question about any of the math that I've done so far or the conclusion that we came to? Okay, let me know if any question comes up later, but I'm going to go on to experiment B. It's going to be similar process. So in experiment B, there's a solution of 10 grams of soluble starch in 100 grams of water. That's added to side A. And we're assuming the molecular weight is going to be 63,000. And an equal volume of water is added to side B. So again, 1,000. So now they're asking us what will happen to the concentration of water and starch, two sides over time. So it's going to be very similar to the last problem that we did, but with different numbers and different molecules. So we're using starch this time. So again, there's 10 grams of starch. in 1,000 grams of water. And they're giving us the molecular weight. So like last time, we're going to divide the 10. And we know the molecular weight is going to be grams per mole. So that gives us 0.00016 moles per 1,000 grams of water. And again, from previously, we learned that 0.1 mole gives us a solute potential of negative 0.23. So again, we're going to do the same thing where we multiply that. So it's going to be negative 0.23 times 0.0016, and that's going to give us negative 0.0004. And on the other side from the problem, they told us that it's pure water. So they said an equal volume of pure water is added to side B. So from that, we know that it's 0 because pure water will always have 0. And it's going to move from a higher water potential to the side of the starch, which has a lower water potential. It's in the negative zone. So we know that water is going to move, but compared to the last problem, we know that this process is going to be a lot slower because, so we got our negative 0.0004, and then our original was 0.69. So it's going to be 0.0004, and that is 172.5 times slower. And where we got the 0.69 is from the last problem. So in the last problem, we found out that this was the um, potential for sucrose, and then the starch is 0.0004. So even looking at the number, we know that for starch, it's very close to 0. Negative 0.0004 is very, very close to 0, then negative 0.69, right? So just by looking at that, you already know that starch is going to be occurring a lot slower just because the difference isn't as big. But if the difference is really big, then water is going to move a lot faster just because the difference in their potential is a lot higher. Did that make sense? Or any questions regarding this problem or the last thing I said? Question asks, is this because the molecular weight is larger in starch? Yeah, yeah. So it's a lot heavier molecule in starch. So that could also be why. It could also be because just the difference isn't that great between the like water without starch and water with starch compared to the sucrose. But yeah, but it definitely comes from the heavier molecular weight because that's what we're dividing it by, and that's what gave us such a small number. So yeah, that's a great question. Okay, so next question, we're going to be looking at fertilizers. So it says fertilizers generally contain nitrogen and phosphorus. And from the other sessions that we did, we know they're required for plants. So the nitrogen is often in form of nitrates, and the phosphorus is in the form of phosphates. So based on what we know about chemistry and water potential, why would over-fertilization lead to the death of plants? kind of think about what adding these things does to the water potential. So adding these compounds, so the nitrates and the phosphates to the soil, so adding a bunch of this to the soil, what that's going to do is decrease, oh, let's see, we have a comment. Creates lower water potential in the soil, which actually draws the water out of plant. Yes, that is the perfect answer that I was looking for. So yeah, adding a bunch of this to the soil is going to decrease the water potential in the soil. And like the comment says, if you add enough, it can even like suck the water out of the plants. So generally, that's why they advise you not to over add fertilizer in the soil. So I'm going to just write that down really quickly. Yeah, so generally this causes water to not flow into the root, but like the comment says, sometimes it can even suck water out of the roots. Okay, and similarly, we have another question, which is talking about overwatering plants, and we know overwatering plants can kill it. And similar to the last question, why this happens is because so the roots are going to require oxygen for cellular respiration, but if we overwater the plants, we're pretty much like filling all the space in the soil with the water. So the soil is where the oxygen is, but if you put water, then water is going to take up the entire space. So without that, there's no oxygen in the roots, and without oxygen, the plant's going to die. So let's see, overwatering equals filling of the air spaces in the soil. And then no air means no oxygen, and no oxygen means cell death. I'm just going to put dead plant. Okay, but here's another question. So if we know that overwatering kills the plants, but can anyone tell me why we can grow, we can sprout roots from cutting the stem in the water? So why is it that we can't overwater soil, but then we can sprout roots in just water? Because like you would think that if we put that much water, it's going to kill the plant, right? But why is it not that? Why can we actually sprout roots in water? Comment says because there's oxygen in the water. Yes, that's the right answer that I was looking for. So, oxygen in the atmosphere can very easily diffuse into the water in a jar or a vase, especially if it's left open out in the air. So, the oxygen in the air can diffuse into the water. So, if you cut the stem in the water, then it's actually going to contain enough oxygen to allow cellular respiration to continue in the roots. Also, like if it's like a clear jar, like a glass jar, and like you leave it like nearby a window, photosynthesis can also occur because you have oxygen and then you also have light and water, so it's very possible. Okay, so next question is xylem cells are dead when functional. Why must phloem cells be alive when functional? Comment says, so you're saying the soil prevents the atmosphere oxygen from getting into the water in the saturated soil case. So in the saturated soil case, what I was trying to say is that there's oxygen forms like air pockets within the soil itself, so that allows for the oxygen go from the soil to the roots. But when you overwater the soil, the air pockets don't really exist anymore because it's just filled with water now. So it's harder for oxygen in the atmosphere to diffuse to that water because it's underneath the earth. So it's like inside the soil. So atmosphere oxygen can't diffuse into that water, and there's also no air pockets anymore. So it decreases the oxygen that the plant has access to. Did that make it a little more clear? Or do you still have questions? Okay, awesome. Okay, so we were looking at question 6. So yeah, why must phloem cells be alive when functional? Can anyone think of why? It has something to do with concentration gradients and sugar. Comment says it requires ATP. Yes, perfect answer. Good job. So the phloem cells are going to be loading sugars against the concentration gradient, right? So anytime you do something against the concentration gradient, it's going to take energy. And to do that, they must be able to use ATP, and only live tissues can do that. So therefore, they have to be alive when they're functional. So phloem cells load sugar against concentration gradient. This requires ATP. Okay, next, we're going to be looking at the forces. So in conduction, the major force is negative partial pressure. So this is what's going to allow them to be moving from one place to another. And it's generated in the xylem. So how that's being generated is there's going to be transpirational loss of water from the leaf surface, right? And it can happen in other places too, but the leaf is the main area on the plant. So when it does that, there's going to be a negative partial pressure. So that's how the negative partial pressure is generated in the xylem. Okay, so next we're going to be looking at the full phloem conduction. So we just looked at xylem, and in the phloem, the major force is going to be positive force. Specifically, it's going to be the hydrostatic pressures that are in the cell. So that's the major conduction, or sorry, the force of the conduction. And how this hydrostatic pressure comes about is so there's difference in water potential in the phloem compared to the xylem, right? And sucrose is being actively transported into the phloem. So this potential difference is also going to pull the water out of the xylem and into the phloem. And this is what's going to create the hydrostatic pressure, and that moves the substances into the phloem. So the force is generated in the water potential. So it's the difference in the water potential. Okay, so the next part is referring to a diagram in activity 34.1. If you guys have done that, feel free to refer to that. So it's talking about the different types of structures in a plant, and we're going to be looking at how are they related to photosynthesis, water conservation, food, and water transport. So first one is going to be the stomata. And when they're open, what it's going to do is it's going to let carbon dioxide enter and oxygen vapor to exit the cell. So the stomata is in the leaves. So it's this water loss that's happening. So when it's open, you're losing water, and when this water loss happens, that's what generates the transpirational pull on the xylem, like we talked about before, because we were talking about the transpirational loss of water. So this is where it's happening. It's through the stomata in the leaves. So but if the plant, say, doesn't want to lose water, say there's not enough water available or it's like super hot outside, then it can close the stomata and it can reduce the water loss. But the downside is that it also is going to reduce carbon dioxide because that's how carbon dioxide enters. So, and plants need carbon dioxide, so that's kind of the downside to that. Okay, and then in that activity, there was also the xylem vessels and the phloem CV. So these are in close contact. And what they're going to do is they're going to facilitate the movement of the water from the xylem to the phloem to produce the hydrostatic pressure for the movement. So this is the phloem conduction that we were looking at. So we talked about the force that's generated. So that's happening in these structures. So these structures are what's helping with the phloem conduction. Okay, so essentially what we did was in the last two parts, we looked at the xylem and the phloem, and we talked about their conduction. So how they move, what happens in there. So in question 8, what we basically did was look at what structure is responsible for that. So if you need to refer back, this part is for the structure. So if you need to know the structure that helps, question 8 will be a good reference. Okay, so question 8. Scientists have measured the circumference of a tree at 2 a.m. and at 2 p.m. If they collect measurements when the ground has adequate moisture and the days are sunny and dry, they find that the circumference, and therefore the diameter, of the trunk is smaller at 2 p.m. than at 2 a.m. From your knowledge of the mechanisms of water transport, suggest the reason for this decrease in the circumstance. So here, on bright, like sunny days, the transpirational pull is going to mainly be due to evaporations from the leaf, right? And it's going to be so great that it kind of like collapses in on like the walls of the xylem because it's really hot outside. So the water keeps like sucking out of the leaf. So it's just so much pressure that it like collapses on itself. And one example is like when you're drinking from a juice box, like if you like suck very hard, then you know how the box kind of like squishes together and like collapses on itself. Essentially, like that's what's happening to the plant. So that's why it's smaller at 2 p.m. when it's super hot than at 2 a.m. So there's an inward force that collapses the sides of the tree, which results in the decreased circumference. Comments asking, do you need to log in to get the survey link for attendance? I was here but just logged in. Yeah, no worries. So I haven't actually put up the survey link yet because I'm just going to do it at the end. And once I put the link, I think I'm not sure if you have to be logged into your PDX email, but as long as you like put in your information, I'll know who you are. Okay, so question question 9 is kind of talking about coming up with an outline of an example. So the experiment would want us to determine how fast the substance is carried in the xylem and in what direction the substance flows in the xylem. So they want how fast and what direction. And another thing is what percentages of the solutes in the xylem at various distances away from the leaves or the roots. Okay, so because it's outlining an experiment question, there's like multiple ways to design this. But I'll just propose one and we can follow through that. So we know the xylem is dead when it's functional, right? Unlike the phylum. So as a result, like what we can do is insert like measuring devices into it, and that's not going to destroy the xylem's functionality. Um, So what we can do is like place these like measuring devices like in 10 centimeter intervals, like straight from the leaves to like the other end of the branch. And so this is like talking about an experiment that's already been done. So what they did was they inserted these like temperature recording devices at intervals along the branches, and they heated the water in the xylem at various locations and recorded how the temperature of the water changed with the recording devices. So they have like water that's really hot, and then they have temperature measuring things along the plant. So measuring the temperature is going to tell them where the water is traveling since the water's hot. So that allowed them to like determine like um, how the water moved, and what they found out was that it actually moved vertically. So they saw that the temperature changed vertically, so instead of horizontally. So they were like, oh, okay, so water in the xylem moves vertically. And then they also took the time that it took for the water to go from one measuring device to another measuring device, and that's also how they found out how fast it went in. Um, and then if we wanted to determine the amount of solutes in the xylem, uh, we can follow the same thing with the like 10 centimeter intervals. And then you can open the taps like periodically and like remove some of the fluid, and that helps you analyze like the fluid in the different parts for their compounds. So that's going to tell you like the percentages of various materials. So I just described like an experiment that's already been done. It was by Bruno Huber. It was like one of the classical studies to determine the direction of the xylem. But now there's like multiple ways to design and test this, but this was just the very classical study that was done. So I'm going to try my best to outline that here in words. So for direction, it's going to be using hot water. Insert temperature probes, and then once you pour the water, see what direction the temperature changes in. And then for how fast, So similar setup, but we just see how long it takes for the temperature to change between one measuring device to another. Okay, so we're almost at the end of our session today, so I don't think we'll be able to go over question 10, which was going to be another like theoretical, like we're gonna do this, but what would you expect? So feel free to try that on your own, and when the professors put up the key, compare your answers to see how close you were. But that was it for today's session. Let me know if you have any questions, and I just put the extra credit form on the chat. So everyone make sure to fill it out to get extra credit. Yeah, no problem at all. Thank you everyone for coming and for interacting in the chat. That makes it more engaging. All right, so it looks like we don't have any questions in the chat. Oh, so can you repost the link? Yeah, of course. There you go. Yep, no problem. So comments asking, how do we find out how many we went to? Um, you can email me. I think that's if you email me, I can count and let you know because I have a track of all the forms that you filled out. So here's my email. So yeah, email me with your name or your, well, your email will be there. But yeah, email me with the name and I can email you back with the information. Yeah, Levin Koala, feel free to email me as well. I can tell you. Okay, any more questions regarding the extra credit or the content today? Okay, everyone, I'm going to end the session. Thank you for coming. Again, make sure to fill out the extra credit link. Thank you. Have a great rest of